Thank God, because Jake did on Monday night and it was awful.
In the car?
No, all over the couch.
Oh, even better.
All right. Where to begin today? There's a couple of things that I want to make sure to talk about. But today's main topic is going to be modes of antibiotic resistance. And we're going to use this as kind of a springboard for lectures on Friday, Monday, and Wednesday, where we're going to talk about three specific examples of antibiotic resistance, or how we can basically reverse antibiotic resistance. Okay? So many lectures for each of those are up. There's not much reading in the textbook per se for these ones. Actually, next week, Wednesday, before class, there's a TED Talk you should watch for that one. And then the test is next week, Friday. So we're coming up to another round. Heads up, this is traditionally the most challenging one, because it is some of the information that is the hardest of the semester. So that's kind of a prelude to get cracking. I know that you guys have had a number of stuff going on with A&P, and we had the practical in here last week as well, but now's the time to start thinking about prepping for this exam. We'll leave it at that for right now, but I'll have some more comments later. All right, what I wanted to start off with then is what we finished. Yeah, Megan.
Sorry, is the exam, you said next Friday?
Yes.
So it's not on Monday?
No. I had to, so you may be looking at what the original schedule looked like. I had to push it back because we had Easter break, and apparently I have to give you guys days off for that, so. Yeah, so it is, so the plan, and I'll make sure that I've updated that. I thought I had, but I will double check Blackboard to make sure it's the updated version. So the updated version has us doing antibiotics today, 3 lectures of microbial evolution, exam on Friday. Okay, so a week from this coming Friday, we've got one more. And then all we have left is final/exam 4. So I wanted to finish off with what we talked about on Monday, which was looking at this this disc diffusion assay, and looking at antibiotic effectiveness on a clinical strain or clinical isolate of Klebsiella pneumoniae. We kind of left off with this last part that we've got something odd going on here between these different antibiotics. So between these two and between these two, there's something going on. And the question was, what is that? What does that signify? So what I want you to do to start off today, what I would like is for everybody to pick one other person to talk to and tell me what's happening here. Why do we see this observation that we've got a zone of inhibition, but yet there's what appears to be another one in between? What's accounting for that? Okay? So give me a description of what may be happening. One of the things you may want to think about as you're looking at this is review what those antibiotics are and how they are the same or how they are different from one another. All right, I'll give you a couple of minutes to try to work this one out. So what's happening? We've got 3 antibiotics here. We have amoxicillin and clavulanic acid, cefotaxime, and then the third one is, I think, our aztreonam. What's happening between cefotaxime and then augmentin, and between augmentin and aztreonam? What are we observing? Combination of the 2 antibiotics. And what's the effect of that combination? Stronger or weaker? Which one? They're working better together than they are individually, right? So this is what this displays what we call a synergy. or synergism. In the case of antibiotics, what we can find is that in some cases we get synergy between two antibiotics, that the combined effect of both of them together is better than the sum of either one's individual effect. And actually, one way to kind of note this is, remember what's true about these disc diffusion assays. Where's the concentration of the antibiotic for Astreonem the highest? On the disc itself, and it diffuses outward from there. This one, I think I wrote down that one had a zone of inhibition of 15 millimeters on its own. Amoxicillin and clavulanic acid has 23 alone. But look at this. That zone of inhibition, all of a sudden, this area here where the two of them overlapped with each other, has a very large zone of inhibition. And so what this allows us to do is, or what we find, is that these antibiotics actually can be used at a much lower dose in combination than using either of them individually to inhibit the microorganism. Some people might call this a combinatorial therapy. This is kind of, in some ways, these types of observations have led to antiretroviral therapy for HIV. So with antiretroviral therapy, we often are putting people on 3, 4, 5 different drugs at one time as a way for them to have a synergistic effect or reduce the chance or frequency of mutants that are resistant to all the drugs at once. Okay. Amoxicillin and clavulanic acid itself is actually a synergistic concoction. What do all of these, what is Astreonem, cefotaxime, amoxicillin, they're all members of what group of antibiotics? Somebody said it. Beta-lactams. And beta-lactams target what process? Or what function of the cell? Not transcription. Cell wall synthesis. Block the penicillin binding proteins in particular. Clavulanic acid is another beta-lactam, but it works slightly differently than amoxicillin. Whereas amoxicillin functions by specifically binding to the class of proteins known as penicillin binding proteins, clavulanic acid binds to what's known as a beta-lactamase, sometimes abbreviated BLA, BLA. So we're going to take a little bit of a look into that. But yeah, sometimes in the opposite of synergy, we will see that two drugs together are actually worse than they are in combination. We try to avoid those at all costs, obviously. All right. So let's take a look at then at some modes of resistance, as that's the major topic. We're going to focus on 3 of these in a bit more detail. 2 of them we'll focus on today, and then the third one we'll focus on Friday, and we're going to talk about Neisseria gonorrhea with respect to mutations. But there are 4 major types of antibiotic resistance. What are those types? So what are the mechanisms that lead to antibiotic resistance? Efflux pump. And Emma, what is the function of an efflux pump? Good. So it is a portin or some sort of pump on the outside of the cell that, when a toxic substance, in this case an antibiotic, gets into the cell, it pumps it right back out. That mode of mechanism requires what, though? This is a form of active transport. It requires energy, ATP, right? Good. So that's one mechanism. That's one we're going to talk about in a bit more detail today. And actually, we've seen examples of this. If you remember when we performed disc diffusion assays in lab, I had you guys test wild-type E. coli and this Tol-C mutant. Tol-C is an efflux pump. without it, we saw generally, not in all cases, but generally, those cells were more sensitive to antibiotics without that efflux pump. What's our second mechanism? Hydrolysis of what? What's that? Well, we use water for hydrolysis, but what are we breaking down? Yeah, the beta-lactam ring or the antibiotic. And the beta-lactam is one specific example of that. So this is the function of beta-lactamases. Beta-lactamases are a class of proteins or enzymes that take antibiotics like penicillin and using water breaks a bond to form an inactive substance. In other words, this hydrolysis step is going to inactivate those antibiotics from functioning. Or, we described penicillin and other beta-lactams as being competitive inhibitors. This inactivation prevents them from functioning as competitive inhibitors now. And I think the name, I think it's it's something like penicillic acid. I can't remember the exact name. I'll have to look at my slides. We'll talk about that one a bit more too. What's third? What's a third mechanism that can occur? I know somebody covered this in their voice thread. How do organisms gain resistance to, or how do viruses gain resistance to drugs? What is that? Mutations. Spontaneous. Some sort of mutation, mistake by the DNA polymerase, which alters the DNA, changing the structure of the protein so that the antibiotic can no longer bind to it. This is an example that we'll take a look at on Friday. So mutation to the DNA. So antibiotic can't bind. What's our last mechanism? Yeah, antibiotic modification. And Megan, do you have a specific example for this one?
I just said that it produces enzymes which can activate.
So yeah, and I actually oftentimes I will group I sometimes will group this one with the hydrolysis of the antibiotic because they're both mechanisms of, broadly speaking, inactivating the antibiotic in some ways, right? So there are some enzymes that will cut the antibiotic, break it down, and then there are others that actually add something to the antibiotic to inactivate it. And as Megan said, these are oftentimes going to be enzymes of some form. One of the most commonly known examples, but this is not one, this is the mechanism we will talk not about after this little 1-minute spiel. A common example of this one is an enzyme that's abbreviated CAT, chloramphenicol acetyltransferase. Basically, what that means is it takes a little 2-carbon-like sugar molecule, it attaches it onto the antibiotic chloramphenicol, and now chloramphenicol can no longer bind to the ribosome. So if it can't bind to what it's inhibiting, obviously the organism has gained resistance to that antibiotic. So 2 of these mechanisms here are ones, actually 3 of them, are ones that involve an addition of a protein in some ways, or a protein performing the function. The last one is more of an inactivation or an inability of the protein to perform its original function, which is the mutation mechanism. We also can see the book groups efflux pumps also with just general membrane modification, so potentially changing of the phospholipid content so that it's harder for antibiotics to pass through. And actually, we've talked briefly about how penicillin is an antibiotic that affects gram-positives, but not gram-negative bacteria. Why does it only affect gram-positives? What do gram-negatives have that allows them to be resistant to it? Not only cell wall, but membrane. The membrane itself is, penicillin can't pass through that outer membrane of gram-negative bacteria. So one of the most often explanations for differences between positives and negatives is either the presence of efflux pumps or just the membrane itself limits the diffusion of the antibiotic into the cell. So these are the four major mechanisms. Any questions on the difference between these? Okay. Let's take a look at a couple of specific examples. Which one do I want to start with? Bear with me as I draw for a minute. So we're going to think in terms of a disc diffusion assay. That's not how I want to draw this. I want 3 plates. Okay, so we're going to imagine a scenario where we have a wild-type strain of E. coli. All right, and let's say that we allow these cells to, and then we have 2 different mutant strains. We'll just call them M1 and M2. And in here, there are 3 antibiotics, all on discs. We perform traditional disc diffusion assay. So our antibiotics would be amoxicillin, clavulanic acid alone, or the combination of the 2 of them, augmentin. Okay, so those are going to be our 3 conditions. And let's say that we observe the following. I will draw these zones of inhibition in a yellow goldenrod color. So let's say that this is the, I did that wrong, son of a gun. Bear with me, my apologies. My apologies. Yes, rip out pages of paper in anger and disgust. There we go. Okay, that's what I meant to draw. So 3 results. Wild type is sensitive to amoxicillin. You can see very small zone of inhibition around clavulanic acid. but resistant to amoxicillin sensitive to amoxicillin and clavulanic acid combined. Okay. Second one shows resistance to all three. Third one shows resistance to amoxicillin and clavulanic acid, but when you combine them, now you've got an effect. These can be, mutant 1 and mutant 2 can be explained by two different forms of, two different forms of antibiotic resistance out of that list of 4 that we've seen. What can explain 1 and 2? Okay, so that's what we're going to try to figure out. Let's start with a brief little introduction, though, to what clavulanic acid is. Okay. So just kind of set that aside for now. I'm going to escape that for the time being, and I do want to save you so I don't lose you. Okay. So, this is one of the modes of mechanism that we've already talked about, that beta-lactamases are these enzymes that will take penicillin and break it down into penicilloic acid, right? Okay? And it does so by cleaving or hydrolyzing this carbon-nitrogen bond right here. This is inactive. This no longer can work. All right? And beta-lactamases in general can basically do this to any beta-lactam antibiotic. There's more specificity than that, but just think in general this can do this to any beta-lactam antibiotic. Well, I'll pull up one more picture real quick. It can't do it to clavulanic acid. And clavulanic acid looks a lot like these other beta-lactams. There we go. Here is amoxicillin and here is clavulanic acid. They look similar. They've both got beta-lactam ring structures. Clavulanic acid is not an inhibitor of the penicillin binding proteins like are the majority of beta-lactams. This, similar in structure, is a competitive inhibitor of beta-lactamases. And so, looking back at that example that we saw, if we have penicillin being broken down into penicilloic acid, clavulanic acid, or I'll just abbreviate it CA here, blocks that from happening. Okay? It basically resensitizes these cells to the original beta-lactam antibiotic. Right? I'll tell you that one of those two mechanisms in that image that we drew, either M1 or M2, is this example, where we see this type of phenotype. The other represents something else. So what I want you guys to do with just this information that we have so far is figure out which of those two mutants, M1 or M2, is the one that has a beta-lactamase, that exhibits this mechanism of being, or exhibits this mechanism of resistance. The other one's something else. Once you've figured out which one has a beta-lactamase, I want you to figure out how is the other one functioning. What's giving it resistance. Okay? So I'm going to give you guys quite a bit of time to work on this one. We'll spend maybe, let's start with like 5 minutes, and then we'll just figure out which one has the beta-lactamase. We'll look at the data to support that. And then we'll go back and figure out the rest.
Can you put the 3?
Yep, I'm going to work on that right now and make sure I can get it up in some way, shape, or form. The reaction. You kind of have to, I would say maybe the best way to do this one, if you're trying to figure out this answer, might be to actually write it out and model for yourself or concept map or do something to help you kind of see the flow of logic here, okay? So we know the following. We know that if we have, oh bother, oh it's not on, that's why. We know if we have amoxicillin alone, if beta-lactamase is present, I'll just call it blah. Can everybody see that, or do I need to change colors? Yeah, thanks. Amox blah. Okay, so if we have that, amoxicillin gets broken down. So we'll just call it, I don't know what it would be called, but basically it's inactive at this point. So it goes from an active to an inactive form. Okay? Everybody following so far? Both of these exhibit that. Both of these exhibit, from the wild type, the cells were sensitive to amoxicillin, and they've gained resistance somehow. Right? So how would we want to figure out which one contains a beta-lactamase? That's where the clavulanic acid comes in. One of the things that we've already said is clavulanic acid is a competitive inhibitor. Apparently I'm dialing the phone. It's a competitive inhibitor of the beta-lactamase. So if you add clavulanic acid, alone it does nothing. It actually does nothing to the cells alone. But if you add it with amoxicillin at the same time, the addition of clavulanic acid prevents the organism from breaking down amoxicillin. And so, in the presence of clavulanic acid, amoxicillin concentrations are higher in the cell than they would be without it. Which of these cells has become resensitized to amoxicillin? So, which of these two is now sensitive again to amoxicillin? M2. And that's how we can identify which one contains a beta-lactamase. Those that have beta-lactamases in the presence of clavulanic acid are going to become sensitive again to beta-lactam antibiotics. The one on the left there has a different mechanism. The addition of clavulanic acid has no additional effect. The cells are still resistant. And so the question becomes, what might account for that maintained resistance even when clavulanic acid is around? Taylor?
Could you use like a mutation or antibiotic modification to stop the acid from stopping the beta-lactamase?
Definitely a possibility. More so the first than the second one. More often what we find with penicillins and its derivatives, the beta-lactams, it's a mutation, specifically to the proteins that these beta-lactams are intended to bind. So penicillin binding protein undergoes some form of mutation that allows it to still function as a protein that's going to create the cell wall, but no longer allows for amoxicillin to bind to it. That oftentimes is what we observe. Okay, so spontaneous mutation, most likely, what kind of mutation would it be that contributes to this change in amino acid sequence? Yeah, it's most likely a missense mutation. That's often what we see. Right. There is another option as well. It could be that there's an efflux pump, that the bacteria have somehow gained an efflux pump. It's a possibility, but that mechanism is actually quite rare. More often than not, the easiest explanation for most of these observations is mutation. And we can identify that. So there are ways now, and this is not something that we spend a lot of time talking about in this class, but we can actually sequence the genome of the penicillin binding, or of those bacteria, and identify, is there a point mutation occurring in the penicillin binding proteins? Okay. One of these represents a mode of vertical gene transfer. The other actually represents a mode of horizontal gene transfer. So mutations again, spontaneous mutations are vertical gene transfer events where one generation to the next, the DNA-dependent DNA polymerase just happens to make a mistake at some point, rendering a subpopulation of cells resistant to the antibiotic. To gain a whole new gene, this doesn't just come out of, well, I guess it does come out of nowhere, but it's not as if E. coli can build this gene on its own. Genes aren't created in a bacterial species. They have to come from somewhere else. This likely represents a mechanism of horizontal gene transfer. So when we see the addition of new genes, like a beta-lactamase, or addition of like chloramphenicol acetyltransferase, ones that are involved in modifying antibiotics, most likely explanation is if it's not inherent to the microorganism to begin with, it's been gained through some mechanism of horizontal gene transfer. All right. I'm going to pause there and just see if there are any questions with that explanation. Aliona?
No.
You need to mull over it some more, don't you?
No, so when you're comparing the M1 and M2, it's
Yeah.
just the one that has a larger zone of inhibition is the one that has the beta-lactamase.
So this one, yeah, this one, basically that zone, I'm kind of trying to draw this in a way to say like this zone of inhibition here is the same as what we find in the wild type. So the addition of clavulanic acid has basically resensitized these cells to what we find in the wild type state. Megan.
I don't know if this is a dumb question, but where did the clavulanic acid come from?
What do you mean by that? Maybe I'll ask that
How is it
first.
just, like, there?
So, I mean, it was lab created, I guess is the first way to state it. It was lab created and it was identified as being an inhibitor of these beta-lactamases. It would be something that we would add during a treatment. So, if, like, for example, you observe somebody has Klebsiella pneumoniae, it's resistant to amoxicillin. Actually, we'll go back to our example and look at that in a second. But then it's not resistant to augmentin. Augmentin is the trade name for amoxicillin and clavulanic acid. That's the name that's given to this combined drug. So if somebody's resistant to amoxicillin, but the treatment the treatment with augmentin clears the infection, you can assume the organism had a beta-lactamase. Yeah. Okay. Yeah, so that's kind of where it comes from. It's one of our options for treatment, basically. Actually, let's take a look back at that real quick, because I don't remember off the top of my head if that example actually had one or not. Ah, not exactly. We've got ampicillin and then amoxicillin and clavulanic acid here, but so a little bit different. Okay. The last thing that I wanted to just take a few minutes to look at is actually visit an example that's similar to what we saw in lab. That one right there. So, and this actually becomes a great precursor for something that we're going to talk about on Monday next week. Okay, I did do this the right way. So here's some data. This is a paper that was looking at Pseudomonas aeruginosa, common microorganism, very challenging to treat though because of a number of different features of it. One of those features is an efflux pump. So the data here, what you're looking at are a range of different antibiotics, 10 to 15 different types of antibiotics. This column here looks at wild-type Pseudomonas aeruginosa. And what they're listing is the minimal inhibitory concentration. This column represents a derivative of those wild type cells. They have deleted a protein known as OPRM. Okay, so OPRM is removed from these cells. In general, what happens to the minimal inhibitory concentration when OPRM is deleted? What's that? It's decreasing. In other words, are they becoming more sensitive or resistant to the antibiotic? Other way around here. Other way around. So if it takes less to treat, they're more sensitive. So what would we conclude from this? If they're more sensitive without OPRM, what might we conclude? Is OPRM important for resistance? Yeah. This is an outer membrane protein. If it's located on the outside of the cell, and if it's contributing to resistance, it's likely that it's a antibiotic efflux pump. Sometimes the other term that you'll see associated with these is the acronym MDR, which stands for multi-drug resistance pump. Okay. So given the last little bit of time we have then, let's take a look at two more pieces of the same data. Here's another wild-type strain of Pseudomonas aeruginosa. Here's its matching pair. Now they're overexpressing OPRM. Okay? So what I want you to do is do two things here. You can, if you're already taking a look at the data, fine. But what I'd like you to do is, if we overexpress this efflux pump, what would you expect to observe to minimal inhibitory concentrations? And then the second part is, does the data fit with that? Okay, so take a couple of minutes to look at this. No, neither of those are mine. Right? Thank you. Thank you.
When you say overexpress the efflux pump, do you mean like the efflux pump is pumping out more?
Yeah, so overexpress, when we overexpress something in bacteria, what I mean by that is like we put in an extra copy of the gene so that you get even more of that protein than you normally would. So you have even more efflux pumps on the surface of the cell. So what would you expect to happen then? So would those cells be more resistant or more sensitive to the antibiotic? So if they're pumping out it more resistant, we'd expect them to be more resistant. In other words, if they're pumping out faster, means that no matter how, if we're throwing more antibiotic, it's moving out.
Do they just one pump? So like you're saying pumps out more and don't pump out.
They all would function the same way. By more, I mean quantity. Yeah.
So that because efflux pumps this side of this, they pump out the antibiotics, not the bacteria.
Yeah, so they're on the bacteria and they're pumping out the antibiotic from the bacteria. Think of antibiotic. Yeah. All right, so just to finish up this conversation, so overexpression means we're putting in basically an extra copy of the OPRM gene. In other words, we're forcing these cells to express, instead of 100 proteins of OPRM on the surface, we're forcing them to express 1,000. With more of those proteins, would we expect them to be more resistant or more sensitive to the drug? More resistant. In other words, what would happen to the MIC? It's going to go up. Do we see that? Yeah. So from 0.5 to 2, that's a 4-fold increase. 0.1 to 0.8, that's an 8-fold increase. 256 to 512, that's another 2-fold increase. And again and again and again. In each of these conditions, what we observe is these bacteria, by overexpressing or by expressing more of this OPRM protein, they're more resistant to the antibiotic. Okay? All right, last comment. I'll stop on this part for today, and then my last comment for today. I have to admit, coming into class today, I was rather surly and frustrated. And let me tell you why for a minute. I know you guys are working your asses off, okay? I know you are. And I know a lot has been going on lately. There are high expectations in this course. I understand that. Some may call it unreasonable. Understand that too. That's just, my kids would complain about it all the time. But I want you guys to come prepared. Okay? That's the one thing that that's the biggest thing that I'm asking you guys to come here with. We can work out the details. This we can work out. I'm not expecting you to know this information before coming into class. I'm expecting you to know the basics. Okay? So what I mean by that is read the book, watch the mini lecture, do some of the homework. Okay? And with that, I think that it's going to help out our conversation. I actually am kind of surprised that today went as well as it did. I was going to come in and pull up the syllabus and say, remember what we talked about at the beginning of the semester. Okay, but you guys have redeemed my faith in humanity for the day. So, good job. Keep it up. I'll see you guys in lab tomorrow for pilot study.